Dr. James Aylward is the inventor of Picato, an anti-skin cancer drug developed from a folklore remedy that has been used to treat more than 1 million patients. A story with humble beginnings, James's journey is the tale of commitment, perseverance, and ingenuity.
I was a CSIRO scientist for 17 years. I had to try and play my trump card, which was a weed in the garden that we'd cultivated in the family for quite a number of years as an anti-cancer agent, we thought, for the treatment of sunspots. So I said, well, I'm going to check this out for sure and try and apply high-level science to examining this and to isolate the active principle if there is one.
Jim's a quiet achiever. He really hasn't had much recognition, and I just felt that it was important that he did. He took a product from just thinking about it.
Picato is registered in Australia for the treatment of actinic keratosis. Some spots, if left untreated, can turn into more dangerous squamous cell carcinomas. A lot of that pioneering work about how Picato works was done here at QIMR at Berghofer as well.
The impact on patients is very significant because you've got 3 days, 2 or 3 days of a simple cream on the lesion, whereas the alternatives are several weeks with potential scarring. So I think effect on patients is significant.
When I first started off, I had no idea what I was playing with. Was it a protein? Was it a lipid? Was it a peptide? Was it a known or unknown compound? And how abundant was it? A CSIRO wasn't interested in my new discoveries. They were prepared to let me work in the lab as long as I didn't use the phones, didn't use the library, didn't talk to anyone, and paid for every chemical I used. I was happy to do that, so I was doing the subfractionation on the extract at CSIRO, and then at night coming back to Professor Parsons' laboratory to test the new samples against malignant melanoma cells in tissue culture.
What a big step for an inventor to move from being a secure position in CSIRO to taking a step out on your own.
I was desperate. I had my back against the wall. It was tough. My father died of cancer 2 years earlier, so it was a bleak period, but the science was going beautifully well. And I was encouraged by the work and by Professor Peter Parsons and Andreas Serbier at the time. We decided that the way forward was to actually publicly list and get many more people to join the Peplin foray. 2 years later, after due diligence, roadshows, Peplin listed on the ASX, and it was an exciting day in 2000, and the share price jumped about 40% on debut, which was a nice thing. It was one of the first biotech companies to be on the ASX.
That's really impressive, and that's just something that I felt needed some award.
The Australian science community are wonderful. They are world-class in what they do. The infrastructure that's been developed here is very special. And without that infrastructure, it would never have occurred what happened that I did. And the other thing we must remember is that most of the innovation does come from basic science. So if you don't have a pool of basic science going, you won't get these little gems of potential translational research.
It's encouraged researchers in the labs here now to do much more in the way of commercial research. And there's a whole program of research based on the protein kinase C inhibitors.
Winning the Clunies Ross Award for innovation would be a humbling experience. It'd be a wonderful thing. It also is de facto recognition for the scientists and the team that was behind Peplin, the scientists and the managers and everyone involved in the Peplin project.
If we're trying to build a vibrant community of scientists and researchers and inventors that all work together, collaborating, then you need to have those success stories held up there as models for what you can do.